Okay, so recording. Okay. So right now we're just going to go ahead and jump into the oral presentations. And to save you guys, 20 to 30 seconds, I would read your, I'll introduce you. And remember, when you hear the timer going off, it means that this group has 1 minute left. And our first speaker, oh, and then at the end, we're going to have question and answer. So we've built in about 20 minutes for that. And the question and answer is, somebody has a question right now. Why don't you put it in the chat box so you don't forget what your question is? And at the end, if you don't get your question in, we will, because we've only allotted 1 or 2 minutes at the end of each of our talks now, and maybe you don't get that in. So at the end, we'll see if we can catch any of those that didn't get answered, or maybe somebody thinks of after the talk. Okay, so our first speaker is going to be Steve Blumenshine. He's at Fresno State, and the title of his talk is The Influence and Impact of Water Allocation for Salmon Restoration. Steve, it's all yours.
Thank you, David. So do I just verbally say advanced slide, right?
Yeah.
All right. You could tell by my title, this forum is probably not used to the type of research I do in my group and with my collaborators with state and federal agencies, but we're interested in the production of a delicious natural resource that needs water. How about that for an ag group, right? So we're working within the San Joaquin River Restoration Program, and I'll draw your attention to the figure in the upper right. So this multi-agency, multi-year program is to restore the Chinook salmon run in the San Joaquin River, which is the southernmost extent of the Chinook salmon range, indicated in purple, all the way from central Alaska down to here in Fresno. So the two main goals of the San Joaquin River Restoration Program, indicated in the green box to the left, is to first of all restore a self-sustaining Chinook salmon population, some good inroads there so far, but also doing that by reducing or avoiding adverse water supply impacts. So given the geographical location of this population and water demand in California, this is quite a challenge. Click, please. So our primary research objectives, and this has been the case for a number of years with this restoration program, is how much or how little water is needed to meet these objectives. So can we grow salmon in less water than you'd think to avoid these adverse impacts? And for that, focusing you on the lower right figure, we focus on the early life history stages of the salmon, the ones that are in the river, like the fry, the pard, and the smolt. Next, please. So I think this group knows some of the issues in California, particularly in the San Joaquin, in the upper center. The San Joaquin River water is way overprescribed. Everyone's screaming for more San Joaquin water. And then on the right, we could look at a drought map that was recently produced. And we see that, no surprise, in the Western US, we're in a very drought-prone area. That's probably not going to change very much. And then in the lower left, this is kind of looking at our salmon landscape. My colleagues from Alaska, you should see the looks on their faces when I show them this in sessions, at fish conferences, but this is what we're working with. We can't restore back to something else, hydrologically or habitat-wise. Next, please. So identification of the problem, one issue is water temperature. We're in such an engineered system, which is undersupplied with water. And in this figure, I'm showing the timeline for when the juvenile salmon are in the San Joaquin River and the temperatures. So it's like a Goldilocks effect. The stippled lines are drought years. Okay, then we're getting towards some critical periods when the fish may not do so good. But in the very wet years, that's why there's a gap, 2011, 2017, there's so much water it stays cold. And the Goldilocks effect would be, we need something that's just in the middle or just right, for example. But we're at the whim of the water management and the type of water years. Next, please. So just to give you an idea of the remarkable effects that turning the dials on the Frying Dam, which holds Millerton Lake or the reservoir, and it releases water in the San Joaquin. I'm showing these images from right near Fresno. So in the upper one, this is with 870 CFS coming out of Frying Dam. That picture was on April 30th. Nice flooded floodplain area, really good nursery habitat for baby Chinook. And then we go there 2 weeks later on the bottom when there's 300 CFS coming out of the dam, and it's a sandbox. Fish don't do well in sandboxes. So we have this very dynamic habitat, not only in area, but in temperature. Next, please. So just another little example of this. We're looking at two images of the same place And the one on the left has a LiDAR simulation overlaying. And this would be the amount of habitat we would have, including the depth patterns of the channel at 1200 CFS coming out of Friant. And on the right is a Google Earth type image with 350 CFS. So we have these and many other tools to look at what happens to the habitat for the juvenile salmon when you spin the dials on Friant Dam. Next, please. Okay, so to make the connections here, and we were asked to talk about the importance of the problem to California agriculture or renewable resources, we're focused on that water. So for our ARI funding, the last 2 cycles for campus funding, our overall conceptual model starts with the blue. Blue is water, of course. So we have the water allocation of the San Joaquin River Restoration Program. That was part of a 16-year negotiated legal settlement between water users groups and the conservation side. So we want to use that water efficiently, moving to the middle blue box, because if the water that's needed for restoration is used efficiently, then there could be more water for other uses. Remember, I showed that the San Joaquin River is way overprescribed, so we need to keep that in consideration. Linking to the green or salmon part, wait a minute, I have 5 minutes according to mine, but so we need to look at what the salmon need given that amount of water to help address the efficiency of water allocation. Okay, next, please. So basically in our 2 rounds of campus funding, the first part with just finishing is generating empirical data. And the next part we're starting now is using simulation tools to look at if you let more or less water into the river, what's going to happen with the salmon. Next, please. So go ahead, we could skip through this because it's just kind of a schematic.
Steve, you still have 3 minutes left, almost.
Yeah, right. Okay, thanks. I just saw the done word. I don't know if that referred to the meal. Okay, so we are looking at the thermal environment, the prey energy, a real bioenergetics focus. Next click, please. And so we're moving from point 1, we're into number 2, or simulations. Another click, if you would. And we're using a lot of simulation tools for that. Next, please. Okay, so this is wrapping it up, take-home results. So in the San Joaquin River Restoration Program, the settlement was designation of 6 different water year types. You can see those on the left. And one thing we've done over on the right here is during the prior drought is to do simulations with the different water level year types, like left to right, critical low, critical high, dry, normal. And my the zoom window is covering up part of it, but the red, blue, and green bars are different amounts of prey in the simulation of prey that'd be in the river. And what we see here, if you look across, the red, green, blue lines are pretty consistent across the water year types, showing us that the fish growth rate, which is our measure of how they relate to the environment, is pretty invariant to water amount, but more prey amount. So this means that if there's enough water, they could do pretty well with low amounts of releases in the tough water years, almost like drought conditions. Next, please. In future plans here's our current objectives for our current funding. We're working with DWR on drone images to characterize the habitat. And we're using an in-stream model, which is a habitat and fish model together. And then working with the U.S. Bureau of Reclamation to estimate fish growth rates and how that varies in time and space. And that's it for me. Hope we got about maybe 1.5 minutes for questions, perhaps, according to my clock.
Yeah, Steve, you actually have 1 minute and 45 seconds.
That's what I got. Thanks.
Great job. Steve, I don't see any questions, so I'm going to ask a question for you real quick. So it's really interesting that you had that consistent trends across there. And I'm wondering, when they start cutting back the water like that, is there a way to canalize portions of the habitat to maintain just a small fraction of it instead of spreading it all out over the entire riverbed or its natural channel?
That's a great point. That would be an engineering feat. That there's other engineering priorities in the river restoration program more downriver. That's a point I'm going to bring up with the fish managers who I meet with regularly. But the river is fairly wild. They let it flow where it's going to flow, path of least resistance and all, and it's a fairly sandy bottom in much of the area that we're studying, actually.
And if I could ask one more question, because there's such a demand on our water, as you say, it's so oversubscribed. Can you kind of guess what the long-term prognosis is maybe for maintaining the salmon populations?
It depends
How do you
on
feel
what,
about it?
yeah, well, feeling is one thing, but science is another, right?
Okay. Yeah,
Um, it depends on the allocations and that comes down to government and
yeah,
who's running the water board, doesn't it?
yeah. Yeah, and then just for everybody, kind of to Steve's earlier point, so ARI definitely funds natural resources. If it's something that you can harvest and control and manage, then that falls definitely within the purview of our ARI charge.
In one bigger picture, David, because we here in the Valley, we see a lot of black and white. Fisher farms, there's billboards everywhere. And I'm advocate, I go out in the community and I say, there's a lot of gray area. Let
Yeah.
us do the science of the fish and no one wants to take all the water. We could help advise on how to have more for homes and farms and other uses.
Yeah, and I think that finding common ground is something that people are increasingly embracing and knowing that that's the way forward. Yeah.
Thank you.
Yeah, great talk, Steve. Thank you. Our next speaker is going to be Sita Sistla. She's from Cal Poly San Luis Obispo. The title of her talk is Plastic in Agricultural Soils and Its Biological Impacts. Are biodegradable plastic mulches a viable alternative? Sita.
Okay, thank you, Dr. Stowe, and thanks everyone for coming today. So yeah, as Dr. Stowe said today, I'm going to be talking about some ARI funded research that began last year, looking at the impacts of biodegradable mulches as an alternative to conventional plastic mulches, specifically in strawberry cultivation, but it's also used in other specialty crops here in California and elsewhere. And so you can go ahead and advance the slide. So I just want to take a quick moment. So this project started as an ARI project, but has actually grown to include collaborations from a number of different groups, including Washington State University and the California Marine Sanctuary Foundation, who are helping to do trials of biodegradable mulches up in the Watsonville, Monterey County area. And so I'll be focusing on data today from Cal Poly, but we have active work in that area as well. Next slide. Okay, so I personally I was a graduate student in California at Santa Barbara. I saw a lot of strawberry cultivation, saw a lot of cultivation in general across the Central Coast. I did not think about how much plastic is used in cultivation of strawberries and other specialty crops until I started to really dive into this project in the last couple of years. And so these are some images that were taken from my graduate student and research assistant last week up in Watsonville when they were harvesting some patches that have biodegradable mulch treatments applied to them. And what this is showing is an image on your left, which is irrigation drip tape being removed at the end of the strawberry season. And the truck on your right is showing the plastic mulch being pulled off of the rows of strawberries. And so for those of you who are not in strawberry growing regions of California or elsewhere, when strawberries are grown, they're in raised beds that are covered with plastic, both for fumigation and then for growing the crop to reduce weed infiltration and increase soil moisture and temperature regulation. And so plastic is an integral part of strawberry production, and it is a big business in California. It's the most profitable specialty crop in California right now, and plastic is a major commodity that is used in this cultivation endeavor. I haven't encountered a single commercial strawberry farmer that hasn't switched to using plastic or a plastic alternative in their production, in their strawberry production. And so just to give you some context for it, in Monterey County alone, about 10,000 acres of strawberries take about 7.8 million pounds of plastic per year. That's disposed of and put into landfills. And so as you can imagine, as this industry has grown in plastic culture, or the use of plastic in agriculture has grown both within California and nationally and internationally, this becomes a huge pollution burden that grows year after year after year. So this material is largely non-recyclable and largely non-reusable. So it's trucked off to landfills. Next slide, please. So there's also, of course, a human component to this agricultural process. So again, when we think about agriculture and the harvesting of crops, the human endeavor in that is often focused on picking the actual crops, but removing all of this material from fields at the end of the growing season and then replacing it at the beginning of the next growing season is also a labor-intensive process. And so there's an economic component in terms of the labor involved in having to literally pull fragments of plastic that get left behind in fields, as well as bundling up these plastics and removing them from the field. So there's a labor cost both in applying and disposal of these materials. Next slide, please. Okay, so this is like a seek and find. This is a little fun game. So what I want you to do here is take a look. I know it's small on some of your screens, and try to find what the unifying theme is in the 4 pictures. So take a second, and then you can click. There's 4 clicks in the slide. Click, click, click. Click. Yes, Susan got it. So plastic. So this is all plastic debris. So there's an irrigation line there in the first picture on your left, but there's also debris embedded in there as well. And so this is all plastic that is left behind in fields, either after cultivation is done or just from previous season's cultivation. So farmers in general try to be good stewards of their lands. They don't want plastic residues to build up in their soils, but unfortunately, when you leave these materials on the soils for months to over a year. So we have one trial up in Watsonville where the plastic will be on the soil in the rows for over a year because the cultivation, the production is still going strong into October now, almost into November. The material starts to break down. And so when that plastic gets pulled at the end of the season, bits and pieces of it get fragmented and they get stuck and embedded in the soils, especially if they're heavier, clayier soils. Next slide. And so this is the overall motivation for this project. Like why think about biodegradable materials? Because I hope I've convinced you that plastics are a major part of agriculture in California and globally, and they are a contaminant in California and globally. And so plastic contamination, while less talked about in a terrestrial or land sense than in waterways, marine and freshwater systems, is potentially a huge environmental burden. It's estimated now that plastic contamination ranges from 4 to 23 times greater than in freshwater systems. And that's a huge range, and I realize that, but that just speaks to the lack of sort of detailed quantification of our land plastic pollution burden. Click. And so it's really only fairly recently recognized how extensive plastic pollution on land and in agricultural landscapes are. And so there have been a number of papers that have come out that are documenting that plastic is accumulating in our precious agricultural soils, and that microplastics in particular, and nanoplastics, as these materials break down from larger fragments into smaller microscopic and submicroscopic pieces, may be actually accumulating in our soils and causing deleterious effects biologically. So they accumulate in animals, they accumulate in plants potentially. And so this is very, very, very new research that's coming out in the last few years. And there's a growing body of research across the world speaking to this idea that plastic in agriculture is building up in agricultural soils. And this is both from mulches as well as other non-point sources of plastic. So for instance, fertilizers that have plastic pellets embedded in them. Next. And so one of the projects that my lab has been working on, and this is not going to be the main focus of my 9-minute talk today, is looking at macroplastic buildup in agricultural soils. And so we've been studying farms across California, looking at the buildup of macroplastic fragments, which is an understudied component of plastics in agricultural soils, and trying to understand what are the biological effects of plastics in agricultural soils when they're large, when they're bigger than 5 millimeters. And what we're seeing, just to give you a preview for next year's ARI, is that it dramatically alters the soil microbial habitat and appears to create a novel habitat for soil microbes. But because of this, and because of other evidence that plastic is deleterious in soils, we want to really understand what's happening, and are biodegradable mulches a viable alternative. Next slide. And so our focal sites are looking at biodegradable mulches. So this is a starch-based plastic polymer as an alternative to conventional polyethylene plastic. And we've done a field trial from October 2020 until August 2021. And we're starting our new field trial this month here at Cal Poly. Next click. And then we have some other trials that are going on up in Monterey County, a couple of different fields that are in various states of being removed from their fields right now and starting again. Click. And we also are studying a specialty field up in Santa Cruz that has used biodegradable mulches for years. They do specialty herb growth. But today I'm going to just focus on a little bit of data from our Cal Poly fields. And this is an image of our students planting. And so we looked at a range of different characteristics, both above ground plant characteristics and soil characteristics in biodegradable mulch applied versus conventional plastic rows to see if BDM, biodegradable mulch, could be a viable alternative to conventional polyethylene in strawberry growth, with the ultimate goal of trying to reduce the plastic waste burden in agricultural production, both in California and elsewhere. And so we assayed strawberry production. We wanted to see how yield compared between polyethylene and normal polyethylene, plastic, and biodegradable mulch. And then we looked at a suite of different soil properties, everything from decomposer substrate preference of how decomposers are behaving to the quantity and quality of carbon and nitrogen in the soil, water content, etc. Click.
Yeah, wrap it up. That's 30 more seconds.
Okay, go. Okay, you can click. And we did a biodegradable mulch decomposition study. So we saw, to get to the punchline, we saw that yield is totally comparable.
Boy, what happened there?
I think I was muted. So our yield was
Yeah, I don't know what happened.
between the biodegradable mulch and the polyethylene mulch. Next slide. And durability of the materials was comparable. And we saw that soil properties were mostly comparable, but interestingly, we saw an increase in available carbon in the biodegradable mulch soil. So underneath the soil, the biodegradable mulch at the end of the growing season, there was more labile carbon for microbial decomposition, which may support and facilitate further microbial activity and further nutrient cycling in biodegradable mulch applied soils. Next slide.
Okay, we're gonna have to wrap it up here. Okay, last slide right here then.
Okay. If you click through, you can just tell. Yeah, and so just to wrap it up, so we found that the BDM and conventional plastic are comparable in terms of their yield and a lot of the different soil qualities, but may increase carbon availability in the soil for microbes. And we're studying right now the potential for offset composting of BDM, because tilling it into the soils is probably not going to be realistic for the cultivation turnover time for strawberry systems. And then we're looking at some of the other long-term effects of plastic buildup in agricultural soils in general. So thank you. Sorry for going over.
Yeah, thank you, Sita. We have several questions for you in the chat. We don't have time to take those right now. You could either answer them directly, and if we have any time left over towards the end, maybe we can open it up for discussion. But otherwise, take a look in the chat. Great stuff. I even had some questions, but I'll hold those as well. So thank you for your presentation. Our next speaker is Katie Tarrant. And the title, she's from Fresno State. The title of her talk is Final Thoughts and Poultry Industry Recommendations on a Journey Through Space and Time. Katie, go ahead.
Hi, thank you so much for having me today. Slide. So I'm going to be talking about this project that we are wrapping up. Can you hear me okay?
Yes, we can.
Yeah, okay. All right, slide, please. And before we do that, I'm gonna start with a couple of definitions just so we're on the same page. Slide. So a broiler chicken is a meat type chicken. These are the chickens that we're consuming. And this project is specifically based off of fast growth broilers. So these are broilers that are bred to be harvested between 6 and 8 weeks of age, as opposed to our slow growth broilers, which take a longer time to age, to processing age, or even laying hens. Slide. So I'm going to talk about identifying the problem. Slide. And before I do that, one more definition, and that is animal welfare. So animal welfare is how the animal copes with its environment, and there's a number of different ways that we can evaluate this. Slide. We know that animal welfare is an incredibly important piece to animal production, and that is not just in reference to how we are managing those animals, but also in consumer buying choices. So consumers are definitely becoming more vocal about what they want, and they want transparency in the welfare of the animals that we're raising. Slide. With any production scheme, it is important to remember that any changes we make on the management side will result in increased costs for our consumer. Now, typically, or I should say for this project, we are specifically concerned with the conventional broiler market. So that is going to be a market where the producers are meeting the minimum requirements for broiler production in the US. This does not include production schemes like organic or pasture raised. So our conventional markets are very important because they provide a nutritious and low cost animal protein to diets across the United States. And it's important that we remember that the price that folks are paying for this conventional market are very are on the low end, and it's very important that we do not raise prices so we price folks out of this particular market. Slide. We definitely always need to be looking to improve in the area of animal welfare. Slide. But we need to ask ourselves, how can we do this sustainably when we think about the price point of our conventional markets? Slide. For this particular project, we are looking at how we can advance welfare within conventional markets by taking practical approaches that are reasonable when we consider cost to the consumer. And we are doing it based off of making recommendations that we've come to through science or through a scientific endeavor. Slide. So an environmental enrichment is a structure that can be added to the bird's environment that helps those animals to display natural behaviors. So an enrichment can be used to help increase animal welfare within a production system. However, they typically aren't used for conventional fast growth broilers because what little research has been done has demonstrated that fast growth broilers just aren't interested in these types of enrichments. Slide. So for this particular project, we are looking to better address that. Slide. Across the project, we're looking at 3 objectives, looking at platform enrichment construction, figuring out how we can implement this on a large-scale basis in commercial houses, which typically house 20 to 50,000 birds in a single house. And then finally, looking at the financial outcomes to using these enrichments. Slide. I'm going to give you a rapid fire of a very small piece of the results that I find to be incredibly interesting, and I hope that that can resonate with you. Slide. But effectively, we created a bunch of different enrichment designs. This graph demonstrates on the x-axis we have day of age for those birds up to 6 weeks of age. On the y-axis, we have a frequency of use of these platforms. And what you're looking at is how traditionally platform usage and thus its value has been evaluated for poultry species of all types. Each of those rows indicate a different design type. And what you can see throughout is we have a general trend of decreased usage over time with these enrichments, which would lead us to believe that these enrichments lose their value over time, which could lead us to saying that platforms in a conventional broiler market is not an effective way to help increase welfare. Slide. Now, the most important point to the results that we have found, in my opinion, is we have reevaluated the way that we measure frequency of use. So we have adjusted that formula, and I could talk about it later if you're interested in it. But what we have shown is that not only are we increasing usage, so we are maintaining the novelty of those enrichments over time, but we have demonstrated that there are specific designs that have huge usage compared to other designs. So our value is not lost in these platforms. Slide. And ultimately, different designs equal a different variation in the usage. With that, we can say that these enrichments could be low-cost options to helping increase animal welfare within conventional broiler facilities. Slide. So a couple of recommendations to wrap things up. First, fast growth broilers require that we do research to understand their own needs. And we don't just apply enrichments because it works well with laying hens or works well with slow growth broilers. Our fast growth broilers have different ergonomic needs, and they have requirements that are different from these other industries. So it's important that we evaluate them on their own, and that we're also sticking to the science, and we are doing things because the birds are telling us that they need it to happen, not because we are trying to apply our ideals on what we think those animals should need. Slide. We have a number of different things that we're wrapping up currently in the future plan section of this grant that should be done over the next few months. Slide. And so with that, I'll just go ahead and acknowledge several folks who have been a part of this grant, as well as our collaborator, Foster Farms. And with that, I'll take any questions.
Thank you, Katie. We will monitor the chat. I don't see anything in there right now, but I do have a question, if I may. What I was wondering, your research was showing on boilers, and I was just wondering, is there just one genetic variety of boilers that everybody uses, so that this is applicable to anything in that generic category of boilers?
So there are a number of different genetic lines of broilers. There are two main providers of these birds, and then within each of those companies, Cobb Van Tress and Aviagen, they have a number of different genetic offerings that focus on feed conversion or efficiency, live weight, yield, meat quality, depending on the genetic line that you decide to purchase and raise. The birds from this particular project were from two different genetic lines, one from Aviagen and one from Cobb Van Tress. So it spans the genetic spectrum, if you will. However, they are very similar in their body structure, their average daily gain, their FCR, and what their ultimate live weight is. So it's our opinion that the results here would be applicable for specifically fast growth broiler lines.
Right, and then you're
also
taking weight to see how weight gain goes over time as well on those different platforms.
Yes, and there are a number of other things that we have to evaluate, like health. So we would assume that using an enrichment like this would be, across the board positive for these birds. However, there are other things we have to take into consideration. Fall slips, breast blisters, hawk burns. There are a number of different health parameters that we have to evaluate to ensure if we are going to use enrichment, we are doing it in the best, most healthiest way possible for these birds.
Perfect. Great talk. And you came in on time. Thank you. So we do, Susan Pheasant has a question for you in the chat, which you could reply directly to her in there. So thank you so much. Our next speaker is Erin Questad. She is a professor at Cal Poly Pomona. The title of her talk is Precision Restoration Treatments for Post-Fire Wildland Ecosystems in Southern California. Erin.
Thanks, David. Next slide, please. Research in my lab is focused on wildland fire-prone ecosystems and understanding how to implement management and restoration practices in these ecosystems. Next, please. And the main issue that we address is the degradation of these ecosystems after fire. Many of them never recover to their natural biodiversity or ecosystem function, and they become invaded with invasive species that continue the fire cycle and their degradation. So we're interested in using management and restoration specifically to try to move these degraded states back towards something more natural. Next, please. But there are many barriers to ecosystem recovery after fire. One is desiccation stress that really reduces survival of plants following fire. And this type of desiccation stress is even more prominent in Southern California, which experiences periods of drought that can make planning for restoration really challenging. Next, please. The other issue that is common now with really large fires is they occur over very large areas, often contain really steep, unforgiving terrain. So it's very difficult to get out and make field assessments in many cases to see what's going on the ground. Next, please. And then a newer threat to restoration is there have been a couple cases in California where pathogens were transported from nursery produced seedlings out into wildland ecosystems. Next, please. And then we're able to infect stands of existing vegetation, causing massive dieback. So there have been really great phytosanitary processes introduced to native plant nurseries, but those are all increasing the cost of restoration as well. Next, please. So our research group takes a precision restoration framework modeled after precision agriculture, where we are using geospatial data to model the landscape. And in our case, we're looking at areas that may be more or less stressful to plants as they recover. Next, please. We developed this framework in fire-prone shrubland ecosystems in Hawaii. And now we are attempting to apply it here in Southern California. Next, please. By introducing more variables into our landscape modeling, such as aspect. As we know, south-facing aspects in the Northern Hemisphere get more solar radiation than north-facing aspects, and they tend to be hotter and drier places in the ecosystem. Next, please. The other part of this project is to rigorously test a restoration method that was developed by the Forest Service as a way of dealing with that issue of nursery-borne pathogens being introduced to the wild. So instead of growing seedlings in a nursery and transplanting them, the Forest Service started experimenting with these plastic tree shelters, which are usually used to put around a tree seedling after it's planted to protect it from herbivores. But they were using it as a way to kind of grow plants in the field by just seeding directly into the shelter. And there have been many studies of these shelters showing that in addition to the herbivory protection, they also create a nice microclimate for plants to grow and get established. Next, please. So in our study, we're working in the copper fire scar in the Angeles National Forest, and we have 30 plots in this fire scar that vary in their environmental stress as we've modeled it using topographic data. And at each one of these 30 plots, we've implemented 6 restoration treatments and are following them for several years to one, look at the cost benefit analysis of these different treatments, and also to see if certain treatments work better than others in certain types of habitats. Next, please. So this is just a little bit of our data to show you what we have so far. These graphs are just showing treatments for seeded restoration treatments. So the green are the seeding into the tree shelters. The yellow is exposed and not protected. And then we also used a cage treatment shown in pink, which was to just separate the protection from herbivores with from the additional protection the shelters provide by changing the microclimate. And these graphs show the proportion of surviving seedlings on the y-axis. And on the left side, it's shown by different slopes, and on the right side by aspect. And those arrows on the bottom are pointing to some interesting results. So overall, our shelter treatment had higher survival across almost all of the conditions that were sampled, except for in the lower stress areas, which would be low slopes and north-facing aspects. But if you look at those arrows on the bottom right, you can see the shelter treatment on our very exposed south-facing slope was almost, the survival was almost equal to the exposed treatment on the more favorable north-facing slope. So by using that shelter, it improved survival to the degree that aspect also affects survival. So we're really excited about these results. Next, please. And we're continuing to monitor the experiment. Next, please. We're also looking at cost. So this table shows the cost of those 3 seeded treatments expressed by the number of live seedlings that were present in each one of those treatments. So you can see that the shelter on the top was the least expensive treatment per seedling. Next, please. And the last phase of this project is to compare these seeding methods with traditional nursery outplants. And so our initial look at the cost was to compare seeding into shelters with transplanting those nursery seedlings out into the field. And outplanting is more than 7 times the cost of seeding into the shelters, mostly due to the large amount of watering that has been necessary because it's been so dry. And we'll be monitoring the survival of those plants through 2 more years to look at the overall cost of all of these different treatments. Next, please. So just to summarize, restoration success in general was lower in the more stressful sites, so steep slopes and south-facing slopes. However, the tree shelters appeared to improve restoration outcomes in almost all of those conditions, and they were much less expensive than other methods. So it highlights this method as a potential cost-effective and rapid and fairly easy way to expand wildland restoration following fire. Next, please. I just want to briefly thank all of the collaborators on this project over the years who have assisted in various phases of the work, as well as the funding sources from NASA, the ARI, and the National Fish and Wildlife Foundation. Thank you.
Perfect. We have room for one question, and Sue Tonic asked if those tree shelters are reusable to your next projects.
Yes, they are. And we do remove them once the plants start to outgrow them and they don't need them anymore. So then they can be reused.
And it seems like water is key to this whole operation because of a steep slope and then a south aspecting. It seems to be the common denominator might be a lack of water. Yes,
it definitely is. And it's really difficult to irrigate in that kind of setting.
Yeah, boy, that's going to be a tough one though.
Yes.
Okay, great talk, Erin, and we
you.
can monitor the chat and see if there are further questions. And we're going to move to our next speaker, which will be Callie Prescott, and I believe maybe Stephanie Cortman, I believe might also be helping her on this as well. They're from Monterey Bay. The title is Soil Greenhouse Gas Emission and Volatile Organic Compounds from Agricultural Management Treatments that Reduce Pesticide Use in Strawberries on the Central Coast of California. And that is a descriptive title. Very nice. Okay, go ahead, please.
All right, thank you so much. Yeah, so my colleague Stephanie and I will be presenting a subset of our results for this study to you guys today. So what we're looking at is that, just to give you a brief background for the necessity of these agricultural management treatments in strawberries. For almost a century, California cropping systems, particularly in strawberries, have used the fumigant methyl bromide to control soil-borne pathogens. But this chemical depletes the ozone layer, and thus it's been phased out per the Montreal Protocol. We've been working with a national team of researchers to evaluate the efficacy of an alternative practice called anaerobic soil disinfestation, which involves adding a carbon source to the soil to feed the microbial population, tarping the field with TIF, totally impermeable film, and then shifting with hopes that that'll shift the microbiome to anaerobic respiration in order to disinfest the soil of pathogens. And we believe this is happening through the production of volatile organic compounds and some greenhouse gases, which may be toxic to some of the soil pathogens. So like I said, we're presenting a subset of results. The scope of this project is much broader, obviously, but that's our goal today. So next slide.
Hi, everybody. So these are the two sites that we monitored emissions from. On the left is the mother trial, or the UC site, where flatbed ASD was implemented, where the tarp covers the entire flat soil surface over the treatment area. And on the right is Plant Sciences Inc. or PSI, where raised bed ASD was implemented, where you can see how just the formed beds were covered with the tarp and the furrows were exposed soil. Takeaways from this are that the ASD treatment was more successful at the UC site, where the anaerobic conditions were sustained for longer. At PSI, the tarp actually melted into the drip tape and created holes that may have impacted anaerobicity and encouraged the release of gases. Next slide. So this represents the method that we used for sampling. We integrated this in situ gas analyzer called a GasMet with tubing that was placed under the slide, under the tarp, and we measured for a specified period of time and over the full 8-week ASD treatment. Next slide. During the measurements, air is pumped through the GasMet and analyzed via Fourier transform infrared spectroscopy, or FTIR. And what's important about this is that the detection limit for VOCs is 1 ppm, but for greenhouse gases, we were able to use a custom range of concentrations based on previous observations that we had made in ASD systems. Next slide. So first, I'm going to present the greenhouse gas results for nitrous oxide and carbon dioxide, and then I'm going to pass it over to Kaylee to discuss the VOC results. So these are time series graphs of nitrous oxide concentrations at each site. The pattern of concentration buildup was similar across both sites, but nitrous oxide was higher in the raised bed ASD treatments at PSI. And you could see that that pronounced peak that occurred there is much greater than what we observed at the UC site. And that smaller graph in the bottom left corner is just like a zoom in on those data, since they're at the same scale and it's hard to see some of the nuances of the UC site data. Next slide, please. So for carbon dioxide, we also saw a similar pattern across both field sites, where the concentrations were highest at the beginning of the treatments and they steadily declined thereafter. And we can see that the carbon dioxide concentrations were much higher in the flatbed ASD, which we're hoping has everything to do with the enhanced microbial activity there that also led to that treatment being more effective at reducing the pathogen. And now I'll pass it over to Kaylee to discuss the VOC results.
There we go. Sorry, I was muted. So for the VOC results, this is one of the first times that ASD has been monitored. The gases underneath the tarp have been monitored in situ. So our primary goal for the VOCs was to evaluate what was there and to evaluate this method of gas measurement, the FTIR. So primarily what we were looking for is just to get some subsample of the concentration of VOCs, and similar to what you see with the greenhouse gas concentrations, the VOC concentrations at UCSC were much higher than PSI, and there were many more different VOCs that we detected. So we found 42 VOCs that we identified at UCSC with the FTIR in the ASD treated plots. We had some control plots that we were also monitoring. These were uncovered control plots that we monitored using a chamber-based method. And we only identified 6 VOCs in those control plots that were also in the ASD plots. And then conversely to the greenhouse gas sort of trend, we found that VOC production actually peaked in the 3rd week of ASD and then declined from there. So there was a steady increase in concentration for many of the VOCs. It peaked around week 3 and then declined thereafter. Next slide. I wanted to point out that the FTIR method, as we mentioned before, it has a 1 ppm detection limit. That's based off of the current reference spectra we have available. So for this method right now, 30 VOCs at UCSC were above the 1 ppm detection limit. And this is important for PSI later when we look at that. We found both dimethyl sulfide and dimethyl disulfide were detected in the ASD, but not in the control. This is relevant because there have been some previous lab experiments by our collaborators who have found that these compounds may be particularly important when it comes to pathogen suppression. Methane was a very dominant volatile, which makes sense given the anaerobic respiration that's occurring, but it was occurring in extremely high concentrations. Up to 21,000 ppm was our max concentration that we detected. And then something that's noteworthy, we found pretty consistently and regularly and predictably, we found propylene oxide was detected possibly as a byproduct of plastic photodegradation. So that was just an interesting note that we saw. Next slide. So conversely to UCSC, the PSI VOC results were much different. They were much lower. We struggled with PSI maintaining anaerobic conditions throughout the entire process. And we saw that reflected in the VOCs that we detected. And so we detected fewer, just the 34. We didn't have any control treatments to compare. So we're looking at comparing the method at PSI to the method at UCSC and their success, basically. And so like I said, we had significant issues maintaining those conditions. Next slide. And you can see how that's reflected in how few of the VOC concentrations reached above that 1 ppm detection limit. And what that means that they rarely were above that detection limit, which means that a lot of the ones that we saw that were below 1 ppm, we can't reliably say or draw conclusions with those concentrations, which is not the case for UCSC, where they were much higher, much more robust concentrations. And then on top of that, we did not detect dimethyl disulfide at PSI at all. And we only detected it once at a very low concentration below that 1 ppm detection limit. Next slide. And when we went back and worked with our collaborators to look at the crop yield for those strawberry crops that occurred, we found that PSI, which is not an organic field, it's an industry non-organic field. We found that the summer ASD treatment didn't do as well as we had hoped. There was disease and significantly lower yield. Or not, I shouldn't say significantly lower yield, but there was lower yield than what we expected. I take that back. Yes, there was significantly lower yield between the ASD and the fumigant. Whereas at UCSC, it was variable depending on the rotation that we performed prior to the strawberry crop, but there was a reliable expectation of the ASD to do well based off of using the flatbed ASD. The structured bed ASD had a lot of problems associated with it, and we see that reflected both in the yield results and also in the VOCs that we detect. So that's an encouraging set of findings for us to proceed further with this and investigating further which of these VOCs is potentially relevant to pathogen suppression, and also gives us some it validates our method to an extent with the FTIR. Next slide. So our future work that we're planning to do moving forward from here is we are in the process of reattempting the raised bed ASD at PSI, and we're trying to monitor the VOCs from the untreated control, the fumigant, and the ASD treated beds, so that we can have a more direct comparison between those in terms of the gases that are present. We began this work in the fall of this fall, but unfortunately we struggled with the ASD treatment again, maintaining anaerobicity. Yeah. We're continuing to monitor the fumigant and UTC to see just to see what data we can get from it. In addition to the field trials, we also have some lab experiments going. These we're particularly excited about. We're inoculating soil with fusarium, and we have 3 treatments with 3 different carbon sources: rice bran, grape pomace, and mustard seed meal plus a rye species. And we'll be sealing that in a PVC pot and monitoring the VOCs for 10 days in order to stimulate ASD conditions in the lab, and then test the post-ASD pathogen load for each of those C sources, and hopefully perform a metagenomic analysis of the soil before and after the simulated ASD, so we can see how the microbiome is actually changing. Next slide. And with that, we just want to thank the ARI Research Institute for the support that they've provided, and also our collaborators at UC Santa Cruz and the Center for Agroecology and Sustainable Food Systems, as well as our industry collaborator, Plant Sciences, Inc. Thank you.
Okay, great talk, both Kali and Stephanie. Sharon Venice has a quick question that that's over there in the chat, and And she says ASD appears to be effective for pathogen suppression, but what about suppression of beneficial microbial activity? Have you measured overall microbial populations or activity via like the PLFA assays? Sorry about that. There we go. Do you have a quick answer for that? I know you're going to do some metagenomic analysis, and maybe that's the answer.
Yeah, I put an answer in the chat too. Our colleagues on this project are actually looking into just that. Yeah, this is a really big team of researchers all over the US, and there are, well, Mark Mazzola from the USDA ARS was on this project, but he retired, and he had already done this kind of work before. And so he had carried some of that, and now there, I can't remember his name, but another USDA researcher is doing the assays, and they will be publishing those findings, and we'll hopefully be able to present this research with all of that.
It would also seem that the shape of the bed may be influencing those microbial activities as well. I mean, you got a flat bed versus the shape, and that actually may be doing something as well. I'm going to guess.
Absolutely. Well, it
Yeah.
also just influences everything about, you know, the implementation of the treatment and trying to keep that whole area uniformly anaerobic. It doesn't stay that way, so
Yeah,
it's not effective.
It's a huge challenge, ASD.
Yeah.
Yeah.
It's a challenge. It's a challenge because it's more effective when it's implemented in the flat system, but that can be difficult for farmers just due to like operational constraints and needing to access their field. Yeah, but hopefully the work that we're doing here will help validate that if it is a treatment that farmers are interested in doing, that, you know, they do it right.
Yeah, exactly. Okay, great talk. I wish we had time for a few more questions. Maybe we'll catch up in the end here. And again, I would invite people to put questions into the chat and let that be answered as we move to our next one. Our next speaker is going to be Elizabeth Scordato, and she is a professor at Cal Poly Pomona. The title of her talk is Assessing the Potential of Small and Large-Scale Native Vegetation Restoration to Deliver Ecosystem Services and Reduce Grower Cost in Citrus and Avocado Orchards. And another descriptive title, so good. So Elizabeth, go ahead.
Great, thanks, David. You can advance to the next slide, please. So it will come as no surprise to anyone here that global biodiversity is in crisis. This recent report from the UN provoked global headlines like this one, click please, declaring that we are at a crossroads for humanity as Earth's biodiversity is collapsing. And as people interested in agriculture, this is something we really need to worry about, click please. And that's because the loss of biodiversity threatens our food supply. In particular, the loss of biological control and the loss of ecosystem services makes it more expensive to grow our food. Next slide, please. This problem has been recognized for a long time in California agriculture. This paper was published 20 years ago, declaring that biodiversity is critical to the future health of California's ecology and economy. And so one of the defining questions of our time, click please, is really how can we make agriculture more biodiversity friendly, click please, while preserving yields and benefiting growers, because people need to eat and growers need to make a living. Next slide, please. One of the interventions that's been very well studied for improving biodiversity in agricultural landscapes is native vegetation restoration on field margins. Click please. There are several benefits to restoring native vegetation on field margins, including the creation of wildlife habitat. It can attract beneficial insects like pollinators and natural enemies, and it can attract insectivorous birds that deliver ecosystem services. However, growers often have concerns about restoring native vegetation in their fields that include concerns that it could attract pests or compromise food safety, concerns regarding the cost of installation and maintenance of native vegetation. And also we have this problem that we don't really have a great sense of the spatial scale at which native vegetation is effective. In other words, how big does a patch need to be for it to be cost effective in terms of the delivery of ecosystem services? Next slide, please. So this is the question that we and me and this group of excellent colleagues are tackling in our new ARI project that started in January. We are asking if we can strategically restore native vegetation to improve biodiversity and deliver ecosystem services. The area that we are working, click please, is the Santa Clara River Valley in Ventura. This is an agricultural valley that is mostly under avocado and citrus production. And it's a great system for asking these questions because the Santa Clara River is one of the last free-flowing rivers in Southern California. And as a consequence, click please, It has been the target of major conservation and restoration efforts by the Nature Conservancy. The Nature Conservancy owns most of the land along, or much of the land along the rivers shown here by these polygons. And in particular, the Nature Conservancy has focused on restoring riparian restoration in 3 areas. Click, please. These are large-scale, long-term restoration plots. Click, please. Where riparian vegetation has been restored for over 20 years. Click, please. And in these areas, we have extensive wildlife census data. So we know a lot about the insects, mammals, and birds within these restored native vegetation habitats. However, click please, we have absolutely no data on how that vegetation restoration has impacted surrounding agriculture. So we've partnered with the Nature Conservancy on this project to ask 3 key questions. The first is how is biodiversity distributed across agricultural land versus natural habitats in the Santa Clara River Valley? The second is what impact does this wildlife have on crop yields? And the third is at what spatial scale does restored vegetation start to impact crop yields, either from a positive perspective or a negative perspective? Next slide, please. So our approach here is 3-pronged. First, we are monitoring wildlife activity and habitat use across the Santa Clara River Valley. Second, we are restoring native vegetation at a small scale to contrast with the large-scale native vegetation restoration that's been done by the Nature Conservancy. And finally, we are assessing the impact of wildlife on crop yields as a function of distance from those large and small-scale restoration sites. Next slide, please. So we've been focused on 4 sites within the Santa Clara River Valley. The first, click please, is Pine Tree Ranch, which is an avocado and citrus orchard owned by Cal Poly Pomona. And at Pine Tree, we've been doing bird counts and foraging tests. We're doing camera trapping. We're doing small scale native vegetation restoration, crop damage assessments, and insect collection. We're also working at 3 sites, click please, owned by the Nature Conservancy, a citrus orchard, and 2 restored riparian sites. At these sites, we're doing bird counts and foraging tests, camera trapping, and insect collection. So next slide, please. I'm just going to show some of the preliminary data we have so far. First, my master's student, Rachel Backrath, has been doing point counts of birds across the entire Santa Clara River Valley. So not just restoration, riparian and orchard sites, but also scrub habitats, suburban areas and urban areas. And this has allowed us to characterize sort of the overall avian community of the entire valley. Next slide, please. So Rachel's data are summarized here. We're looking at a summary of the dissimilarity indices between the 5 different habitat types. What you'll notice, click please, is that each habitat hosts its own unique bird community. And I want to draw your attention to the orchards here. Click please. The bird community in orchards is most similar to the scrub habitat, but orchards do host their own unique bird community that is characterized by a particularly high diversity of dietary niches. So we have lots of different types of birds eating different types of things in the orchards. Next slide, please. So the next question we're of course asking is, how does this bird community impact crops in the orchard? To get at this question, click please. Rachel is doing preference tests looking at foraging behavior of birds on weed seeds versus native seeds and beneficial versus pest insects so that we can understand how those birds are impacting crops. Next slide, please. We're also monitoring the mammalian community at Pine Tree Ranch and at one of the riparian sites. We're using camera traps to do this. You can see one circled in this picture. And we have found some remarkable things with our camera trapping data. Next slide, please. What we're looking at here are images from just one camera at Pine Tree Ranch in the avocado orchards. We can see a huge diversity of mammals, including bears, mountain lions, coyotes, bobcats, mule deer, skunks, and raccoons. Next slide, please. And so far, we're actually finding, click please, that the diversity and abundance of mesocarnivores and other mammals is actually higher in our orchards compared to the restored riparian habitat. Click please. The activity patterns that we're picking up on cameras suggest that the orchards are part of these animals' home ranges. They're not just transiting through. And click please, conversations with growers suggest that there are no obvious negative impacts of mammals on crops in these orchards. So that's an exciting thing to be finding. Next, please. Finally, we've started our small-scale native vegetation restoration project. We have planted, click please, 3 10-meter by 10-meter plots on Pine Tree Ranch of native shrubs. The shrubs have been chosen to maximize structural variety and floral composition. They've been in the ground for about 6 months now. Click please. So they are finally big enough to start having an impact. Next slide, please. So My student Anne-Marie Martinez has started measuring the impact of these native vegetation on crops. Click please. So Anne-Marie is measuring leaf and fruit damage and assessing insect communities along transects up to 75 meters from these restored plots. As those shrubs get bigger, we're going to expand that radius. Next slide, please. So to summarize what we have found so far, orchards support a unique bird community within the Santa Clara River Valley that is characterized by a broad array of dietary niches. Next. Mammals appear to be more abundant and diverse in orchards compared to restored riparian areas. Next, please. And together, our preliminary data suggests that orchards may actually be really critical to the maintenance of biodiversity within the Santa Clara River Valley. Next, please. In the future, we'd like to expand our monitoring and restoration to additional properties, including both orchards and row crops, so we can contrast different crop types. We'd like to include small mammal trapping to monitor potential mammalian pests, as well as mesocarnivore control. And we, of course, as our native vegetation gets larger, will assess the costs and benefits to growers of having crops in proximity to these small versus large scale restoration plots. So I'd like to thank all of the collaborators on this project, especially our funding from the ARI, and I would be happy to take any questions.
Yeah, we have some questions from the chat, and Dilruba asked, are you looking at both positive and negative impact by birds?
Yes, absolutely. So that is a key question. We want to know if the birds are providing an ecosystem service via pest removal or if they are damaging crops. So we're assessing fruit damage and foraging behavior on beneficial insects versus pest insects.
And then we'll look later to see what kind of ecosystem services. So one of the things that one might imagine is that maybe farmers, growers have to spray maybe less often. Maybe they can cut back one spray or not spray all of their orchards.
That would be a hope, yes.
And it's, I think, also important to note that how much land agriculture controls in our state and nationwide. And I'm going to guess that when you present these results to growers about all the wildlife coursing through their orchards, they're going to be a little surprised, but pleasantly surprised.
I hope so. The growers that we have spoken to have been very enthusiastic about the wildlife on their land, and particularly noted that all this effort has been focused on restoration on the river, but that they have all these animals on in their orchards that seem to be doing great.
Yeah, which are adjacent to the riparian areas, right?
Yes.
So there's a nice corridor.
Yeah, exactly.
Yeah, that's great stuff. Boy, I like seeing all that. So great work, and we'll monitor the chat and for further questions. So thank you for that presentation. Okay, our next speaker, and she will be actually the last for our 9-minute presentations, is Magdalena Maj. She is at Cal Poly San Luis Obispo. The title of her talk is Oral Administration of Coronavirus. We've heard that word lately. Coronavirus spike protein provides protection to newborn pigs when challenged with porcine epidemic diarrhea virus and reduces GM-CSF cytokine levels in sow's milk. Magdalena.
Hello, everybody. Thank you for having me here today. I will be presenting the results of my team of undergraduate students at Cal Poly San Luis Obispo, on which we have worked over the past few years. And to start with, I would like to tell you that the abbreviation which I'll be using during this presentation, porcine epidemic diarrhea virus, will be PEDV to make it shorter and easier. Next, please. So the PEDV is the disease which was first reported in European countries in 1970s and then in Asian swine industry over the last 30 years. And first in May 2013, PEDV suddenly appeared in US swine industry and rapidly spread across the country. And that virus killed within the very short period of time a population of 10% of pigs. And it created a major financial economic loss to the swine industry, about $1 to $1.1 billion per year annual revenue. Next, please. So this virus, PEDV, is transmitted fecal orally. What it means is it's an entering disease which infection is occurring in the GI tract. So PEDV infects and replicates in the cell lining of the small intestine and in the colon. And you can see in this drawing here on the left how the virus is actually entering the enterocyte. And this viral replication inside of the enterocytes is causing vacuolization and destruction of cells, and this leads to this virus atrophy. We see here on the right side the histopathological analysis of the jejunal tissue, which was derived from pigs on the top infected with the PEDV and on the bottom control animals. You can visually see that those villi from pigs infected with PEDV are much shorter. That means there is a villi atrophy, and this will cause malabsorption. So the animals cannot absorb nutrients properly. Next, please. So clinically, PEDV will be causing acute watery diarrhea and dehydration in pigs all ages, but high mortality only in neonatal piglets. So in older pigs, what you can see in those pictures in sow, grower, and finisher, the disease is less severe and pigs mostly get sick. They lose a lot of weight when they are getting infected. However, the newborn pigs, right after they are born and they are suckling the milk from the moms, from the sows, they are usually dying within 5 to 7 days from the moment of infection, because they are lacking in maternal protection, they are lacking immunity, the immune system is not yet developed. Next, please. So the goal of our project was to work on the novel approach to develop an oral vaccine against coronaviruses. Here we are working on PEDV, but the same may actually apply to a current coronavirus SARS-CoV-2. And we have chosen to work with the plant system. So we express the S protein, which is a part of the coronavirus protein responsible for the binding of that viral receptor to the host cell, in that case on the enterocytes in the small intestine. And that S protein is also very important for that entry to the host cell. So we have selected that S protein to clone it and express it in the plant system. The plant system has an advantage because when animals will consume, in our case, we have selected corn, if they will be consuming corn, it should mimic the coronavirus infection because it's entering through the GI tract. So it should be protecting the mucosal tissue. It is safe, it is much more attractive mode of administration as compared to the injected vaccine. It's also helping with the minimization of cost and labor. And also we don't need to use needles. Next, please. So these are the steps which we took in order to develop that oral PEDV vaccine. So first of all, we express the S protein and we prepare the DNA construct. We transfer the DNA construct with the S protein to the Agrobacterium tumefaciens, which is a bacteria known since many years to efficiently transform plants, and in our case, it's corn embryo. Then we have selected the corn embryos that are successfully transformed, and we've grown those corn in the greenhouse. We have identified the corn lines which have been expressing a very high level of that S protein. We prepared the corn or grain for pig studies, and now we have conducted several animal trials. And I will be presenting to you today one of the most important animal trials and how we have shown that this vaccine is working. Next, please. So the objective of this animal trial was to determine whether pregnant sows, which were administered corn-expressing S1 protein, will transfer the lactogenic immunity to those suckling piglets and provide protection against PEDV. So very important to indicate here is that the sows, the moms, the pregnant sows are getting vaccinated with the oral vaccination with the S protein in the form of corn. And then piglets are being born, and we will be challenging actually the piglets with the PEDV virus. Next, please. So this is our study design. We had as a positive control injected commercial vaccine. We had 2 doses of our oral corn vaccine with the low dose of 10 milligrams of antigen per feeding per pig, and a high dose of 50 milligrams of antigen per feeding per pig. We had also a control corn, which was untransformed commodity corn. We have, when the pigs were farrowed, 3 days after that, we have challenged the piglets through intragastric inoculation, and we have collected sera and milk samples on several days. Next, please. One of the most important assays which we have performed in the lab was looking at the neutralizing activities by fluorescent focus neutralization assay. And this is the assay which will allow you to have an idea whether you have the level of neutralizing antibodies against the PEDV virus. Next, please. So here you can see in this graph disease severity in piglets after the PEDV challenge, and the blue line is showing you the piglets which were from moms administered the low dose of the oral vaccine candidate, while the injected one is shown in gray. So you can see that they are pretty similar. Still, the low dose is showing the lowest disease severity index. The control and high dose animals were pretty severely sick. Next, please. And this is the most important finding, which is showing that the percentage survival of piglets from different groups, and you can see that there is the highest survival of piglets in the oral vaccine with the low dose, so they reach at 55% of the survivability, while in the positive control, the injected vaccine, it reached 37%. Next, please. And here, one of the analyses which we did for the level of those neutralizing antibodies in the milk, and this was done post-challenge, specifically 6-day post-challenge, we can see that there is a high level of neutralizing antibodies in the oral low dose PDC vaccine. And this is confirming that the sows were providing that passive lactogenic immunity to suckling piglets. Next, please. So the conclusion from this animal trial is that corn, which is expressing S1 administered to pregnant animals at that low dose of 10 milligrams of antigen per feeding, provided the lactogenic immunity and protection against challenge with PEDV virus. So I would like to acknowledge my industry collaborators, ABI and Mazen. Great thanks to Agricultural Research Institute and helping us with that study, which really allowed many undergraduate students to get involved in the both animal work, animal trials, as well as a lot of laboratory work. And also thank you to the Calpori Swine Unit. Thank you.
Great, thank you, Magdalena. We have time for maybe 1 or 2 questions. Do we have any in the chat? I will monitor that. In the meantime, while we're waiting for a question, I'll ask, so your sows were fed the transgenic corn only during the pregnancy, and I was wondering if they were continually fed it even while they were lactating and nursing the little piglets, and if they were, would that help raise the antibody levels and maybe even the survival rate?
This is a very good question. So it takes some time. So maybe we can go to the next slide. I don't know if Lauren has that slide. Yeah. So it takes some time for the if you look at those green molecules, this is showing the PEDV virus, but the same will be representing our oral vaccine. So it takes some time for that immune response to kick in. you have to represent this antigen on the DC, on the dendritic cells. You have to activate the B cells, which are then migrating through the blood. This is like 0.5 on that slide, migrating to the mammary gland. So, and then you have already these plasma cells producing specific IgA. So this is the antibody produced in the milk, in the mammary gland, and then in the milk. So I don't think it will really be that helpful because those plasma cells are already there. And there is no evidence that continues in that case within the 7 days continuous administration of the vaccine would be really helpful because we already have those plasma cells producing IgA.
So that's actually good news. Feed it to them once and you're done. So basically, okay, that's great work. And we'll continue to monitor your chat if there are any follow-up questions. You know, I always have other questions, but I'm too busy to put them in the chat because I'm limited on how many things I can concentrate on him. But you know, everybody, that was really great work, and in all of them, I would love to hear more about it. So thank you for everybody's excellent presentations. And without taking a break, because your cameras will be off, so you can take a break anytime you want, but we're going to jump ahead and go into the lightning talks. And, you know, I'm hoping that everybody is able to hear right? You can hear that. So unfortunately, I'm going to be doing that at your last 20, that's when you hear that you got 20 seconds. And I've instructed Lauren to, at 3 minutes, to go ahead and advance to the next speaker. So we're just going to kind of rapid fire go through this. Write down your chat questions as we go along, and we will do a few minutes afterwards when everybody's finished, and we'll do a Q&A session. Okay, so our very first speaker is Kun Zhang at Chico State, and the title of his talk is going to be Upcycle Olive Pomace as a Natural Additive to Entrance Performance of Asphalt Paving to Enhance, I'm sorry, my writing is so small here, to enhance performance of asphalt paving materials. So this is going to be quite an interesting use of this olive pomace. Okay, Kun, it's your show here.
Thank you, David, for the introduction. My name is Kun, and I'm an assistant professor from the California State University, Chico. And my work is upcycle the olive pomace as a natural additive to enhance the performance of the asphalt paving materials used for the asphalt pavement. While the research motivation from the waste management of the olive oil industry. So during the olive oil extraction, so about like 70 to 80% of the material are byproduct, which is olive oil, and the rest of the 20 to 30% that is olive oil. Well, in California annually, we kind of like produce about 150,000 tons of the olive pomace in California. So that is kind of like issues to do this waste management. Well, globally, especially in European countries, they have 20 to 30 million tons of the olive pomace created. So right now, the current recycling method of the olive pomace is to use that as animal feed or use the soil fertilizer. Well, it may cause some problems, for example, anti-digestive problem for the animal, or the reduced seed germination if it is used for the soil fertilizer, because the olive pomace is acid, pH is lower than 7. Well, we propose to use the olive pomace as an additive in the asphalt and enhance the asphalt pavement because first, the olive pomace is rich in lignin, especially the polyphenols. So this is a really good material for the antioxidant additive, while the asphalt pavement also need the antioxidant additive to resist the UV agent, to resist the thermal oxidation. And the second fact is the olive pomace contains a certain amount of the residual oil. We tested the pulp-rich friction, so it contains about like a 13 or 14% of the residual oil, and this residual oil can be used as a recycling agent for the pavement. So we process process the olipopis in the lab, mix it with the binder, do the binder test, do the mixture test. Well, what we found is this from the binder test is the olipopis did excellent work as an antioxidant to retard the aging of the asphalt. And if we put 15% of the olipopis, so about 25% of time can be extended for the asphalt to experience the oxidation-induced cracking. While for the mixture, yes, it is the olipopis can be used as a recycling agent for wrap materials. So the next phase of the study is we are going to evaluate the mechanism of the antioxidant and also the recycling agent, why the olipopis works, and also run the life cycle cost analysis. With that, thank you so much.
Boy, that's excellent, Kuhn. Thank you. You actually had 20 extra seconds. So I've written down 2 or 3 questions that I'll ask you at the end. Thank you so much. That's great. That's very interesting. Please stick around. I've got 3 questions for you. Okay. Our next speaker is Gabrielle Salada Fink, and she's at Fresno State. See, that's how much time you had left. And does that? There we go. There we go. Sorry, everyone. She's at Fresno State, and the title of her talk is Supplemental Lighting and Carbon Dioxide for Enhancing Citrus Nursery Growth. Gabriella.
Hello?
There we go.
Hello.
No problems. No problems.
I am working with Dr. Grit Brar, studying different ways that plant growth can be or is currently enhanced in nursery greenhouses. Next slide. We are studying plant growth enhancement in citrus by manipulating the photoperiod with supplemental LED lighting. The larger idea behind this is to provide a model in which greenhouse nurseries can maintain or increase plant growth development with less use of propane heating. It's a pretty costly input for both nurseries and the earth. The need for this research stems from the costs associated with developing the insect exclusion facilities as a mitigation effort against Asian citrus psyllid, also called ACP. ACP transmits a pretty serious disease in citrus called Huanglongbing, otherwise known as HLB. Next slide. Last winter, we performed a study with a partner nursery in Woodlake, California. We applied 5 lighting treatments, a control, a day elongation, a night interruption, a full daytime LED supplementation, and a far red daytime supplementation. Next slide. The carbohydrate partitioning analysis from this experiment is still currently being processed, but our initial growth data shows some promising results when the young citrus has already pushed. We're going to go ahead and repeat that experiment this winter with the same task-specific grow lights that we used last year, as well as a more utile, economically mindful option. Next slide. Separately, we're looking at the interaction of high temperatures with elevated carbon dioxide levels in citrus. ECO2 in the context of high heat has been well studied as climate change progresses and agriculture tries to keep up. We'd like to go ahead and investigate levels that may negatively impact young citrus development in nurseries. Next slide. So during our proof of concept experimentation rounds, we witnessed some really interesting data in regards to the rate of growth versus data obtained from the Lycor 6800. These 2 graphs show that the plants kept in the ECO2 environment had higher stomatal conductance and photosynthetic output than the control group, but that didn't necessarily correlate to significant growth over the control group, which was very interesting. Next slide. Sorry, next slide. And that just is going to go ahead and lead us to the next round of questions for this experiment. And we'll go ahead and explore those as we move forward in the next few phases of research, specifically looking at feedback inhibition in the elevated CO2 systems and the nitrogen assimilation pathway. and the regulation of that. So thank you very much.
Perfect, thank you. Our next speaker will be Alexander Lopez, and also from Fresno State.
Nope, it's actually going to be Michaela Gunther.
Oh, I'm sorry,
Hello.
I skipped one. Hello, Michaela. Will be Michaela Gunther from Humboldt. Not even close.
Thank you.
Yeah, assessing conservation impact of leaving slash piles and recently harvested areas. Okay, go ahead, please.
Okay, great. Thanks for inviting me to speak here today. This is a work in progress. Next slide. My goal is to assist the timber harvest managers to evaluate the conservation significance of slash piles. What are slash piles? Next click. Yeah, thanks, Lauren. They're small to large accumulations of debris that are byproducts of the typical timber harvest operations. You can see in the top picture. These are often burned to reduce fire risk, as we know is a huge problem in California, but that causes soil sterilization, exotic species establishment, air pollution, greenhouse gas emissions. So we're interested, next click, to maintain these slash piles as complex habitat for threatened and endangered species, such as this federally threatened Humboldt martin. These slash piles may serve as cover for the prey species, or they may serve as cover and stopover points for the mesopredators themselves in this managed landscape. Next slide. So how are we going to assess the problem? First, we'll put out some trail cameras. We've placed these at the harvested slash pile areas and in paired mature forest stands. We're looking for Humboldt martin and fishers. And we're also conducting small mammal trapping to determine prey availability in these paired sites. And finally, we're about to set the dogs out because they can sniff out and detect scat from a variety of carnivore species. Handlers collect the scat. We then send them to a lab for DNA metabarcoding, where we can establish the diet of the carnivores and compare what's eaten with what's available in the environment. Next slide. So my hope is that we can get some occupancy of the local carnivores, especially for species of special concern in California, for the objective of creating data-driven support for future forest management plans. And we also hope to determine prey availability to get data-driven support for the maintenance of slash piles in these areas. And finally, some professional training. I've got master's and undergraduate students who are working on research theses and publications to train the next generation of wildlife professionals. Overall, what we really want to promote is responsible forestry. It's a huge economy here in California. Last slide. So I've got some great people I'm working with. Wow, my Women of Wildlife graduate student, Alyssa Roddy, undergraduate student, Shalom Fletcher. Next, I've got some great collaborators. I'm working with a landowner, a local private timber company, California and Oregon Green Diamond Resource Company, providing in-kind and logistical support. The National Conservancy for Air and Stream is giving me some match and in-kind support. And finally, of course, HSU, CSU, and the ARI are providing some really important financial support. That's it. Thank you.
Well, that's great. I just have to, we've got like 15 seconds. See very many martins around there? I think it's just totally interesting.
It's so exciting. The Humboldt martin, there may be about 400 remaining on the entire Pacific coast. So we don't see many, but we are seeing them in these managed landscapes. And knowing that so much of our forest is being managed, we really want to be able to support these threatened carnivores in these habitats that are managed.
And the bottom line is you can maintain, there's a balance perhaps between.
Exactly. Yeah. Work with these harvesters and Green Diamond, but really a lot of the timber harvest companies around here have really been working to promote ecology that's good for wildlife. So it's been a great partnership.
Excellent. So I want to again remind everybody we're going to be live and in person up in Sacramento. I hope all of you from this talks today will attend next year so I can engage you in very long conversations and pick your brains and see what you
That would be so fun. I've really missed out. So maybe we can get together next fall. Thank you for having me.
Yeah, that's great. Thank you. Okay, now this time our next speaker is Alexander Lopez from Fresno State. The title of his talk is Origins of Amaranthus tuberculatus, which is also known as water hemp, in Central Valley agroecosystems, a population genetics approach using genotyping by sequencing. See, I saved you 30 seconds by announcing that. Go ahead.
Thank you. Yeah, I just want to talk about my project real quick, looking at water hemp in the Central Valley. Next slide, please. So basically, water hemp is a dioecious summerannual herb. It's native to the Mississippi Valley. There are two varieties. One variety on the western side of the Mississippi is rudis. The other variety on the other side of the Mississippi on the east is the tuberculous variety. And so over the last about half century, this weed has become really troublesome throughout a lot of agricultural ecosystems in the Midwest, particularly damaging in soy and corn, largely due to their ability to rapidly evolve herbicide resistances. Slide, please. And so water hemp's not native to California, and it's not been reported to be problematic, but within the last decade, it's been reported to be invaded agroecosystems here in Merced County. And so basically for my project, we're just kind of looking into where these infestations originate possibly from within their Midwestern native region. Slide, please. And so to do this, we took a population genetics approach where we collected samples from these populations in Merced, sequenced them, and then compared them using genetic clustering methods to potential source populations in the Midwest here on the right. You have the rudis variety here in the red, and the tuberculous variety in the blue. Next slide, please. And so to do this, we use the program structure. Our results in the Midwest clustering largely correlated with previous results where we identified 3 units, a Western unit, an Eastern unit, and then an admixture unit in the ranges that overlap. And then, so what we found is that Merced actually clusters with the Western subunit here in the blue, which has actually been attributed to be the primary driver of the agricultural invasiveness in the Midwest. Next slide, please. And so this is really important to the local ag industry because these Western biotypes are really problematic throughout the Midwest. And this could have resulted in the introduction of pre-adapted genotypes that that could be really problematic here and become difficult to manage. So there's a lot of work to be done to look at these populations. Within our lab, in the future, we're going to be looking at some of the physiological capabilities and assessing some of the herbicide resistances that it may potentially have using genetic and dose response studies, as well as looking at germination phenology and growth potential. That's it. Thank you.
Great, perfect. Different than ragweed though, close relative, I guess. Okay, so our next speaker is going to be Megan Hines from Monterey Bay. The title of her talk is Impact of Arbuscular Mycorrhizae Fungi, Application on Soil and Nutrient Utilization in an Organic Production System, the 2020 Sampling 18S, the 2020 Sampling 18S and ITS1F AMF results. Megan, go ahead.
Great. Thank you. Good afternoon, everybody. I'm Megan Hines. I'm adjunct research faculty at CSU working in the Hoffa lab, and I'll be talking today about the metagenomic results we have for the arbuscular mycorrhizae at our site. Slide, please. So a lot of us are aware of the importance of soil microbes in ecosystems, but in terms of agriculture, there's a lot of questions. For instance, who is there? What microbes are there and exactly what are they doing? And how are they actually interacting and functioning together? And so this leads us into questions that pertain to management. So how do certain healthy soil practices impact those communities? And then even broader questions, as in how do these communities, are they changing with climate change on this planet? And so we selected a site on the Central Coast at a commercial operation that is organic and grows specialty crops here. Slide, please. So we worked in romaine for this part of the study. We have 3 controls, 3 plots that had compost, 3 plots that had compost and commercial mycorrhizal inoculant. We took composite soil samples in each of the plots down to 8 inches and sent them to a commercial lab in Sacramento called Novogene for metagenomic analysis. We looked at 18S as well as ITS1F sequences, and the lab reported back data in terms of operational taxonomic units, which are specific sequences that match up to various species. So what I'm going to show here is just the arbuscular mycorrhizal data, specifically the glomeromycota data. So next slide. So this lists by the sequence, either 18S or ITS, which species we found. So we have several very common species. We had Pheniliformis mossiae, Gigaspora margarita, and Rhizophagus irregularis. So those are very common, especially Pheniliformis mossiae. Those are very common to arable lands. Looking at it with a very, very basic ANOVA here, just to see what was going on. Keep in mind, this is just a survey, not extensive sampling. We found that only in the 18S sequences was a statistically significant difference, but it wasn't among the treatments. It was between the sampling times of July to September 2020. The ITS, we did not see any significant differences there. That's why the p-value is not reported. So you may also be asking, okay, you applied a commercial inoculum. So the product we applied was MycoUp, which is Glomus aranicum variety tenahypharum. Apologize for the pronunciation. So Novagene's results did not say that this was there initially, but doing some research and looking into it, there are some taxonomic synonyms for this particular species. So Rhizophagus aranicus, Glomus aranicum, and Dominicia aranica are all taxonomic synonyms. And looking in two other additional databases, Mycobank and Margem, we found evidence that likely the species is present at our site. However, unfortunately, it looks like it is everywhere. So it didn't just stay in our plots. And with that, I'll thank you for your guys' time today.
Perfect. Thank you, Megan. We'll again circle back to ask questions here at the end. I'm sure people will have some for this one. Our next speaker is Javier Ramirez. He's from Cal Poly Pomona. And the title of his talk is Effects of Feeding Time on Feed Efficiency in Steers. Javier.
Hello, everyone. Thank you for having me. So my presentation is on the effects of feeding time on feed efficiency on steers. Next slide, please. So the beef production industry is valued at $2.63 billion with rising expenses, the main being animal feed. By increasing an animal's feed efficiency by a small portion, it can have a major economic impact. Next slide, please. So in order to tackle this problem, we house 16 steers separated into an AM and a PM group based on weight, 2 steers per pen, 4 pens per group. Each of those groups were fed 12 hours apart. Each pen was fed approximately 7.3 kilograms of hay and 21 kilograms of grain ad libitum. At the end of those 12 hours, any leftover feed was collected, removed, and weighed. Feed intake was collected daily and weight was collected weekly. Some of those calculations that we had to find was average daily gain, which is how much body mass is produced on a daily basis. To find that, we take the initial weight, subtracting from the final weight, divided that by the number of days, which is 133 days. The feed to gain ratio is a measure of an animal's efficiency in converting feed nutrients into the increased body mass. To find that out, we took the total amount of feed of each pen, divided that by 2, which is because of 2 steers per pen, and then divided that value by the initial weight over subtracting it from the final weight. Next slide, please. With that preliminary data, we're able to produce these results. The AM group is represented by the orange and the PM group is represented by the blue. So the PM group actually had gained 1.2 kilograms per day. And then in terms of body weight, they all started with an average of 430 to 434 kilograms, but the PM group ended up with an increasing of 566 kilograms for the final body weight. Next slide, please. In terms of daily food intake, they had a constant daily food intake ranging at around 24 kilograms per day. And in terms of the feed conversion ratio, the AM group had for every 14.7 feet a feed that was consumed, they gained 0.45 kilograms in terms of weight. And then the PM group actually had less. So for every 11.1 kilograms of feed intake, they produced 0.45 grams of weight. Next slide, please. With that preliminary data, we can conclude that the PM group may have a lower feed to gain ratio and have a higher final body weight, concluding that feeding at night may have led steers gaining more weight and consuming less feed, which is essentially what we would like to have less feed intake, but produce more body mass. And that is my presentation. Thank you.
Great. Thank you for that presentation. Our next speaker will be Carly Johnson from Humboldt State. The title of her talk is Biochar Quality Assessment Characteristics Based on Production Method and Feedstock Type. Carly.
Hi, thank you for having me. My name is Carly Johnson, and I work as a student research assistant at the Schatz Energy Research Center at Humboldt State. And the topic for my presentation today is biochar quality assessment characteristics based on production method and feedstock type. Next slide, please. Biochar is a carbon-rich material that remains after pyrolysis of biomass in oxygen-derived environments. It has the potential to provide environmental and economic benefits to California's agriculture sector, such as improvement in soil quality and mitigation of problems related to nutrient runoff and soil contaminants. There are several deterrents to the predicted growth of the biochar market, such as inconsistent labeling schemes, lack of quality assurance practices, and a wide range in prices between products. The goal of this work is to test and analyze biochar to determine the connections between production methods, characteristics, and their recommended application. From this, recommendations will be made to stakeholders that improve the transfer of information from producer to consumer. Next slide, please. This study used 12 unique biochar samples procured from industry stakeholders that include biochar either already available on the market or from byproducts from energy generative processes. The sample's input parameters were categorized by wood type and production method. Wood type was either by hardwood, softwood, or shell, and production method was either by combustion, gasification, or use of a kiln. Some of the important testing completed in-house included SEM imaging, proximate analysis, and sorption and leachate testing. After testing was complete, a NOVA and Welch's t-test were used to analyze the data. Next slide, please. Biochar products derived from softwood yielded the greatest water holding capacity and fixed carbon content. Also, our analysis showed that biochar characteristics are more dependent on wood type than production method, specifically for the variability in pH, fixed carbon, and recalcitrance. The results from Welch's t-test with respect to feedstock type for fixed carbon can be seen in the plot on the right. This plot shows the comparison of fixed carbon content in biochar feedstock types, with the comparison of feedstock type on the x-axis and the dry basis percent mass difference on the y-axis. The p-values, which are included below each box, suggest the softwood feedstock resulted in significantly different fixed carbon content when compared to the shell and hardwood feedstocks. This box plot was created for all of these statistically significant measured variable results by testing if there was a difference between the data points with consideration to both the input parameters. Next slide, please. In conclusion, further analysis is important to improve the biochar market, increase efficiency of biochar usage, and improve the communication between producers and consumers. This work was made possible by ARI and the staff at the Schatz Energy Research Center. Thank you for listening.
Carly, thank you so much. Our next speaker is Rafael Cuevas-Ribi from Humboldt State. And the title of his talk is Evaluating the Growth of the Seaweed Pacific Dulse at Different Depths at Humboldt Bay. Rafael.
Yes, so this is a collaborative project with Green Wave and Karen Gray. And this is the work of my graduate student, Erika Thalman. Next. Seaweed is an emerging agricultural industry in the United States, but we still largely depend on imports. We need to increase the local production to fulfill the growing demand of seaweed. Next. There is a need for California to wake up and be part of this growing industry. Thanks to this grant, we were able to establish the first commercial permitted seaweed farm in open waters. And last Saturday, we presented the results at the California Seaweed Festival. Next. So we want to launch a seaweed industry in Humboldt Bay. Because it's such a new industry, we need to know how to culture the seaweed, when, where, how much can we produce. Today, I'm just going to be talking about the best depths that you can grow Pacific dulse. Next. The system consists of 2 long lines. Each long line is 100 meters. Within the long lines, we have droppers. So every 2 meters, we have a droppers where we evaluate the growth of the seaweed. So we are evaluating at the surface, 1 meter, 2 meters, and 3 meters depth. Next. So we found out that the best growth was at the surface at 2 meters, at 0 meters, and 1 meters. So the seaweed growth at 2 meters and 3 meters was full of biofouling. As you can see on the picture on the upper right, biofouling meaning there is a lot of aquatic organisms that are going to be outgrowing your seaweed. Next. So on our second trial, we were able to produce more than 1 pound in less than 60 days. And this was at 0 meters. And what we found is that if you don't harvest at that time, the current is so strong in the bay that is going to take away that seaweed, that big bundle out of your rope. Next. And that's what we found out. In the first trial, about 50% of the seaweed was lost. But on the second trial, we found that most of the losses occurred at 2 meters and 3 meters. Next. So in conclusion, the best depths are going to be down to 1 meters. The best season is going to be spring and fall. You should avoid growing seaweed on the summer just because there is a lot of biofouling, a lot of aquatic organisms that attach to the seaweed. And as soon as you get a nice grow seaweed, you should harvest or trim it. Next. Thank you.
Thank you, Rafael. Our next speaker will be Juan Vega from Fresno State. The title of his talk is Developing a Mobile Decision Support Nitrate Sampling and Monitoring System. Juan?
Hello, everyone. My name is Juan Vega. I'm a student research assistant here at the Center for Irrigation Technology. And next slide, please. So basically, for our background as to why we want to do this is groundwater pollution is a large problem around the world, especially here in the Central Valley. About 1/3 of tested irrigation water exceeds the MCL of 45 milligrams per liter. And this concern highlights a need for a highly mobile nitrate analyzing unit, which allows for real-time sampling. And through this unit, growers will be able to monitor different areas of a field. Next. As for our objectives, we want to develop a system that could potentially change the practices of growers here in the Central Valley. And again, it will allow growers to monitor their nitrate levels. Also, we want to change this because it will lead to less contamination, and less contamination would mean less contaminated runoff water. And we also want to demonstrate correct application to smaller growers who may not have the knowledge or the resources such as larger and corporate-owned farms. And also, we would also do surveys that were given after workshops in hopes of garnering potential change in grower practices. Next. As for a description of our tower, we have a CR1000 data logger, which is used to record our different measurements, and we also use it to automate the system. 5T sensors were used to measure volumetric water content, temperature, and EC. And then our solid state relays were used to power solenoid valves that were used, and the solenoid valves were used to for both irrigation and fertigation practices. And as for the UMS vacuum, it was used to evacuate sampling bottles to extract our soil water solution. Next. And then, as you guys can see to the picture to the left is our CR1000. It's the black box with the green ports. And then right under that is our solid state relays that would control our solenoid valves. In the middle picture is the bottles that we would use to collect our soil water. And then to the left is the UMS vacuums that are used to extract the water to get into the sampling bottles. Next. And as for findings, there's much technology in other industries, but I mean, it's not really implemented for agricultural use. So that's a pretty big problem. And it was very difficult to adopt because it's very hard to get this technology to communicate with each other. And also the maintenance of technology from the industries that we use is difficult because the technology isn't specified for outdoor use. So we would have problems with getting water in it, or simply the Central Valley heat would overheat some of these technologies that we would use. And that's it for me.
Okay, thank you. Thank you, Juan. Great job. Our next speaker is Sean Carpenter from Fresno State. The title of his talk is The Organic Production of Agretti in Poor Quality Soils with Poor Quality Water.
Thank you for having me. My name is Sean. I work for Gary Banuelos at the USDA ARS, and I'm going to be talking about growing agretti with poor quality soil and water. Next. So one of the biggest problems facing the California ag community is the continued drought we're in right now. The entire state is facing at least some form of moderate drought, with certain areas, including the Central Valley, experiencing an exceptional drought at the moment. So this is producing a lack of good quality water for irrigation. In addition to these water issues, farmers on the west side of the San Joaquin Valley are also dealing with the poor quality soil in the region, specifically the presence of high salt, boron, and selenium, which you can see in the photo on the far right. And there's over 400,000 acres of such farmland available on the west side of the Central Valley. Next. So what we're doing at the USDA right now is evaluating agretti as a new alternative organic crop that can be grown with poor quality water and soil in various drought conditions. And so to do this, we prepare our own irrigation solutions with salt and boron. And we have 3 different application rates. The high, or 100% shown in this table, is basically a non-drought condition where the plants receive adequate water that you would if you weren't water limited. And the medium and low treatments are just 3/4 and 1/2 of that respectively. In order to start this type of experiment, we grow the agretti in seedling trays in our greenhouse. Next. Then we transplant to a field on the west side of the Central Valley. Next. And they're grown here until harvest. Next slide. So far, we have demonstrated that you can grow agretti in this poor quality soil with a minimal amount of poor quality water. The fresh weight yields range from 9,500 to 3,800 pounds per acre for the high and low irrigation treatments respectively. And in addition to the overall yield of the crop, we wanted to look at plant stress and see how this lack of water is affecting them. So to do this, we measured the total phenolics, which are a bioindicator of plant stress. And we've shown that agretti that received less water had an average higher phenolic content than plants that received adequate water. So the plants are being stressed by this lack of water, but they're still able to produce a respectable yield. And keep in mind, this is a crop that you can harvest up to 3 times a year due to its short growing cycle. Next. And last, I just wanted to show you a little bit about what happens with the agretti we grow after. So there is a high demand for it at some of the upscale restaurants in the region, such as Max's Bistro in Fresno. They're always calling, asking for our agretti. And it's also sold in the Fresno State Market here. So not only is this something that you can grow with limited amount of bad quality water, but it is a product that is desired. And just a second before my time runs out, I just wanted to thank the ARI for making this research possible, and everyone at the USDA and CIT. Thank you.
Perfect. Thank you, Sean. Our next speaker is Chantal Stieber from Cal Poly Pomona. The title of her talk is Iron and Nickel Catalyst for Mitigating Denitrification and Agricultural Pollution. Chantal?
Okay. Thank you. Next slide, please. So agriculture uses a range of fertilizers which contain the element nitrogen. Nitrogen is critical for plant growth. However, 50 to 70% of added fertilizers aren't taken up by crops and end up as waste. Agriculture accounts for 73% of nitrous oxide, 54% of nitrate, and 84% of ammonia emissions in the US. Next, please. Our dream is to take these polluting agricultural byproducts and recycle them into new fertilizers. This presents a significant problem for chemistry because small molecules such as nitrous oxide, nitrate, and ammonia are generally challenging to activate or use to make new molecules. Next. Our approach is to use iron and nickel catalysts with N-heterocyclic carbene ligands, and we make these in our lab to study activation of nitric oxide and nitrous oxide. Once activated, we can build new molecules that could then be reapplied as fertilizer. Next. If we take a close look at our catalyst system, we see that there are several ways we can modify the system to change the reactivity. In purple, we can change the metal. So here we're working with nickel and iron. In green, we can tune sterics or bulkiness. In orange, we can change the electronics, and in blue, we can tune the bite angle. So to the right, you see 4 of the possibilities that we're currently testing in our ARI project with the bite angle. Next. Once we've added the ligand to the metal, we react it with nitric oxide and nitrous oxide. We do this in the glove box, which is shown to the right, in the absence of air and moisture to carefully control the chemistry. We then test the activation and reduction in a stepwise fashion to build new chemistry. And students have already activated nitric oxide and functionalized with hydrocarbon sources, and we're currently working to fully characterize products and expand the scope. Next. So in summary, we've made new iron and nickel catalysts. We just received new EPR instrumentation, which was funded through the first year of our ARI grant, that will help us identify the metal nitric oxide, nitrous oxide species. So this is sensitive to unpaired electrons. And we're actively working to identify NOx activation and reduction. Next. So with that, thank you to the ARI, NSF, DoD, LSAMP McNair for funding. You can follow us on Twitter at StieberLab. And most importantly, all the work was done by amazing undergraduate and master students at Cal Poly Pomona.
Great, thank you, Chantal. Our next speaker is Sean Ashcan from Fresno State. The title of his talk is Groundwater Sustainability Agency Irrigation Scheduling Toolkit. Sean?
Thank you, David. Regarding the problem and issue, we know groundwater is a major source of water supply in California, providing 40% on average and 60% on more in dry years. And the Sustainable Groundwater Management Act will bring restriction in groundwater pumping. SGMA will change farming in the future, and this project responds to such changes. SGMA requires each basin to form one or more groundwater agency by 2017, prepare the plan by 2020, and implement the plan by 2040. The California DWR reported the formation of more than 260 groundwater agencies in 2017. As you can see, these agencies cover most of agricultural land in California. So these groundwater agencies are the heart of SGMA to implement the plans. They have difficult job to do, and they need technical help, obviously. Next, please. For the experimental approach, the objective is to help McMullen area with over 120,000 acres. This agency is located in Kerman, just west of Fresno, and irrigation is the most significant part of water budget. This toolkit is designed to help with irrigation water calculations. The specific task of this project include creating 6 different databases: weather, crop, soil, field, crop water demand, and irrigation scheduling databases for the agency service area. Weather database covers all the local weather station. Crop database compiles the data for local crops. Soil database is based on the USDA soil map. Number 4 is the field database that covers all the field boundaries and the cropping pattern in the agency service area. Next is crop water demand, and finally, irrigation scheduling database will be prepared separately for each field and combined for the agency service area. Irrigation scheduling here is simply the amount and timing of irrigation water application to meet crop water demands. All 6 databases will be created using ArcGIS and Microsoft Azure utility. Next, please. Here is an example of database number 4. We prepared this map for the agency, and their technician can use laptops, iPads, and cell phone to verify the field boundary and quickly update the crop info. They can click on any field, make changes in the field or office, and update the changes as needed. Thank you, David.
Perfect. Thank you, Sean. Our next speaker will be Lauren McMullen from Monterey Bay. The title of Lauren's talk is The Impact of Herbuscular Mycorrhizal Fungi. Sorry, guys. Jeez. Fungi application on soil and nutrient utilization in an organic crop production system. Percent inoculation results for romaine lettuce. Lauren.
Thank you. My name is Lauren,
and I'm an undergraduate student researcher at CSU, and I am researching a subset of the project that Megan talked about earlier, specifically the percent inoculation results for romaine lettuce. Next slide, please. There's many benefits to having microRIZE in a field condition. Specifically, it provides increased nutrient uptake and also acquired resistance against plant pathogens, which reduces our need for plant pesticides. So the microRIZE indirectly decreases greenhouse gas emissions and also helps to preserve the health of topsoil. MicroRIZE is also used to assess plant health based on different fertilization techniques, which is how we use it. Many of these benefits are not always seen in agricultural research because there's so much human impact because there's so much activity in the field. But our goal for this is to study the percent inoculation of a mycorrhizal specialty crop. And this is an inexpensive and quantitative way of assessing the plant health. Next slide, please. So we found 3 random plants in each replicate plot. And then from these 3 random plants, we got 3 root samples, and we washed the root samples, and then we stained them with a fountain pen ink. And then we destained them with acetic acid and water so that we could have only the arbuscular structures stained. And then we stored them in lactoglycerol until we were ready to count them. To actually count them, we used the grid line intersect method, and we first counted the root intersections where any of the roots intersected, and then for the next count, we only counted the intersections with the arbuscular structures. And then for our results, we weren't able to actually statistically analyze them yet, but it looks like both the compost and compost plus mycorrhizae treatments seem to have an increasing average percent inoculation when we compare them to the control. Thank you.
Okay, great. Thank you, Lauren. Our next speaker will be Lisa Bentley from Sonoma State. And the title of her talk is 3D Forest, Quantifying Above Ground Carbon Stocks and Fire Fuels to Inform Natural Resource Management. Lisa?
Great, thank you. So my collaborative work here is funded system-wide by ARI, and it's focused on quantifying above ground carbon stocks and fire fuels across these diverse gradients in Northern California to inform natural resource management in light of climate change, right? Just like Dr. Still said. So why do we want to do this? Next slide. So the California Air Resource Board estimates that about 85% of the carbon stored in the state's land base is found in forests and shrublands, as you can see from the green coloring on this figure on the top of the slide. But there's about 20 to 40% uncertainty in these estimates of above ground biomass. So to support the state's climate strategy, accurate above ground biomass estimates are needed to determine amounts of carbon sequestration and then greenhouse gas reduction from timber and natural lands. Next slide. In addition, historical fire suppression and poor forest management has led to a buildup of fuels and increased wildfire frequency and severity. So as a result, land managers need to implement fuel management approaches to improve forest health, but this requires an accurate quantification of fuels. Next slide. So given these issues, we're focused on using a terrestrial laser scanner or TLS and a drone or a UAS to acquire detailed measurements of forest structure. Next slide. Since 2019, we've been collecting these data across 4 sites in Northern California. At this point, we've sampled almost 100 plots. Next slide. Here you can see an example of our plot level data that we collect with TLS, and you can see some of the trees that we can extract from these plot level data. Next slide. And here you can see that we can extract forest structure metrics from our drone or our UAS using structure for motion processing. Next slide. So I'm just going to show you some figures of results very quickly. But in general, we've been very successful at using these approaches to estimate and evaluate forest structure We can use UAS structure for motion to estimate canopy cover and capture changes in the canopy post-wildfire. Next slide. And TLS can also estimate surface fuels and ladder fuels and also above ground biomass across multiple species. We've also been able to use the TLS to create new allometric equations for oak species. Next slide. So as you might have noticed from the citations on the previous slides, we're completing this work with a great team of graduate students. A lot of these publications are in process with them as leads. And in the future and in person at the meeting next year, I look forward to sharing more results from 3D forests and hope that these results that we have from our project will help assist land managers with accurate carbon accounting, timber management, and building ecosystem resilience to reduce risk in light of future wildfire and current climate change. Thanks.
Perfect. Thank you, Lisa. Our next speaker is Dilruba Yisman, and she's from Fresno State. And the title of her talk is Water Smart Planning, a satellite imagery-based remote sensing approach to evaluate crop water status in California.
Next slide, please.
Thank you, Dilruba. Go ahead.
Next slide, please. Public Policy Institute of California actually, in their 2015 and 2018 report recognized that there is a need to manage farm water for the long-term basis. Now, this same report also recognized that, we can avoid expensive field level data collection if we can adopt some advanced technologies like remote sensing or so for this long-term analysis. Now, UAV-based imageries, drone-based imageries are kind of getting popular in agriculture, but and they also provide almost real-time field monitoring, but they're all still limited by space, time, cost, and in some cases accuracy. So in that case, for long-term monitoring, probably it is a nice, try to use the free of cost satellite imageries to use to find overall trend or pattern in the crop water level in California orchards. Next slide, please. So why it is important for California? So solve the water crisis type science we're seeing in California for a while now. So this problem is persistent and it is not going away so quickly. We all can agree on that. So this proposed project is designed to analyze actually long-term water status in California orchards, which will help to find long-term trends and patterns in water status, and also in a space and temporal and spatial scale. It will help to understand how these orchards deal with the wet and dry years, and also overall with the drought. Next slide, please. Okay, so the specific objectives are basically to analyze and describe the trend in water status utilizing multiple decade satellite images. And the second one is how we can, have the policymakers, ag industry, growers utilize this data. Different steps to follow: list all the major crops, collect available satellite images and categorize them depending on certain criteria, perform field validation, calculate different vegetation disease, and find trend in water status level for multiple years in some major orchard crops. Next slide, please. So we have some preliminary results. It's a work in progress. So we found from one specific field validation orchard on our campus farm, Fresno State campus farm, from the pistachio orchard, that there is a difference in value range for NDVI generated from UV-based data and also satellite-based. So why is the difference? And if there is a correlation, we're going to find out. So we are adding more field validation orchards, some elements, and some other crops too from our on campus, and also some other analysis results found that some specific orchards, like in the entire California, showing some positive and negative trends in NDVI. So evapotranspiration data is also we're analyzing to see that if this can correlate too. As for the same date and for the same kind of data for a couple of years difference, like in the right picture, as you can see, 2016 and 2019, same dates, same orchard, but showing some differences. So we're just trying to understand that, with the transpiration. So we're hoping for some exciting results. It shows already showed some positive correlation with the transpiration. So we're going to see if satellite images are really useful for this long-term analysis. And that's pretty much it. Thank you.
Great, thank you, Dilruba. Our last speaker is Haruka Tanaka from Cal Poly Pomona. The title of her talk is Oxidative Cleavage of Lignin Models Using Vanadium Catalyst.
Hello, everyone. My name is Haruka, and I'll be presenting on our project. Next, please. So we have some problems in current society, which are like petroleum and fossil fuels, which are basically the current source of chemicals and energies. And that could cause some environmental concerns. And also agricultural waste are generated a lot each year. So it is always better if it's reused or recycled, because it could cause contaminants and it is being used inefficiently so far. Next, please. So we try to solve those problems using the agricultural waste, which is basically the biomass. That is a renewable source that could be an alternative to the petroleum and fossil fuels. And then biomass, if we could convert the biomass into value-added products, it will be a good way to solve the problems. Next, please. So for the method, the objectives of our project is to create the lignin model compound to produce some utilizable products. In order to do that, we synthesize the lignin model compound and vanadium catalyst, and we study the temperature and ligand effects in catalytic reactions. So for synthesis, next please. So we synthesize the beta-O4 lignin model compound, and the picture on the right side is the sample structure of lignin structure. And then there's a lot of type of linkages, but the beta-O4 linkage is one of the most commonly found type of the bond. So we try to make that beta-O4 lignin model compound to study the cleavage. Next, please. So we characterize the products using proton NMR spectroscopy. Next, please. And for the vanadium complex synthesis, we first synthesize the ligand. Next, please. And then this is the how we characterize the product using the proton NMR as well. Next, please. And then using volatile ACOG and ligand that we synthesize the complex of the vanadium. Next, please. Next. And then we characterize the vanadium complex using IR spectroscopy. Next, please. So for the results, we run several catalytic reactions using the synthesized model compound and catalyst. Next, please. So this is the picture of how we run the catalytic reactions. Next, please. So this is the proton NMR catalytic runs and some of the products. Next, please. And then for the catalyst, we use volatile ACOG and synthesized complexes. Next, please. Next. Yeah, and here's the table of the results. And next. And we found that like electron withdrawing group on the ligand have the highest yields and conversion, and then 120 degrees Celsius tends to have higher yields than 140 degrees for 3 days. Next, please. So for the acknowledgement, I would like to thank to Cal Poly Pomona, CSU ARI for the funding, and Dr. Alex John, who is my research advisor. Thank you so much.
Thank you so much, Haruka. Great talk. I would like to thank everybody for all of their talks today. And if we have any other questions, then we can post them in the chat. You know, I just want to ask one or two questions as long as I see some people that are still here. Lisa, how scalable is yours? If you are working on plots, and I don't know the size of your plots, and we have hundreds of thousands of acres, is any of it scalable, or at least can you extrapolate on the data with a little bit more variables in it?
Sure, that's a great question. I was just editing a paper from my graduate student, Brianne Forbes, who's looked at scaling our measurements of ladder fuels. And so what we measure on a 20 by 20 plot scale basis correlates very well, significantly, with the same type of data that we can extract from airborne LiDAR. So it looks like these measurements will be able to be extrapolated to larger spatial scales.
Okay, good. Let me ask one other quick question. Javier, you know, I've had heard all the, you know, for decades, I guess, that, boy, you shouldn't eat, you know, right before you go to bed. And I'm wondering if you do this PM feeding of cattle and they were, had increased efficiency in terms of weight gain, how did that affect the quality, you know, fat versus muscle?
Unfortunately, we're still in the process of analyzing that data. We did have a couple of the steers actually slaughtered the previous couple weeks ago, but actually in November, we're actually going to be slaughtering them again, the remaining, and then we're still going to be analyzing the marbling and the body fat and the back fat of the animals.
Okay, so we'll wait for those data, and hopefully you guys will be up in Sacramento next year so we can actually look at that data and maybe even taste some of those steaks.
I'll take note of
that. Yeah, put them in the freezer. But I wanted to thank everybody. I think that most everybody has been keeping up with asking questions in the chat box as we've gone along. So thank you again, everybody. One of the things I wanted to point out is that by doing what we just did today, we can see the diversity of the projects that ARI is funding and that our faculty are working on. And I will say that all of our students are doing a great job in the work that you're doing and the presentations that you did here. Over 95% of all of our projects have at least one student working on them, and we showcased a lot of you guys today. And again, fantastic work. You guys are very knowledgeable and doing great work with this. Now, the other thing that I've noticed here in what we're doing is as I'm kind of monitoring the chat box, you guys are doing great jobs networking. So talking to each other and planning future talks and maybe new research projects, but at least now you know what each other are doing. And I know that some of you will be following up with each other and brainstorming ideas about how to combine some of your research, and you probably got some ideas about how to do your own research here as well. So thank you everyone for taking time this afternoon to participate and to listen. And if you have any follow-up questions, feel free to reach out to everybody. Feel free to reach out to my office, and we will see everyone again next year. Thank you. Bye.